Hello, everyone, and welcome back to yet another video. This is going to be the one from biology, and we'll be discussing the first lesson of genetics, that is, principles of inheritance and variation. For the ones who are new here, we discuss NCRT and literally every line of it in the easiest possible way so that you can just watch your videos on the go and you're ready for any exams because mostly our exams are now all about NCRT these days. So, without further ado, let's just get started. So, have you ever wondered why an elephant always gives birth only to a baby elephant and not some other animal, or why a mango seed forms only a mango plant and not any other plant? Given that they do, are the offsprings identical to their parents, or do they show differences in some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other, or sometimes even so different? These and several related questions are dealt with scientifically in a branch of biology known as genetics. So, what do we basically do in genetics? Is we have a look at the variations, at inheritance. What do we inherit? What do we get from our parents as part of heredity? This subject deals with the inheritance as well as the variation of characters from parents to offspring. Inheritance is the process by which characters are passed on from parent to progeny. It is the basis of heredity. Again, very important line. So when it comes to inheritance, basically characters parents से offspring में कैसे जाते हैं उसको inheritance बोलते हैं, and inheritance is the basis of heredity. Fine. Variation is the degree by which the progeny will differ from their parents. So we all know it like is part of basic observations you could see even at your home. Also you can see siblings being so similar, or different relatives being so similar, and sometimes even very close siblings being very different from each other. That is the variation, basically the degree by which you would differ from your parents. Human knew from as early as 8000 2000 BC that one of the causes of variation was hidden in sexual reproduction. So obviously because there's recombination, there's variation. Fine. They exploited the variations that were naturally present in the wild populations of plants and animals to selectively breed and select for organisms that possess desirable characters. For example, through artificial selection and domestication from ancestral wild cows, we have well known Indian breeds. Example, the Sahiwal cows in Punjab. We must, however, recognize that though our ancestors knew about the inheritance of characters and variation, they had very little idea about the scientific basis of these phenomena. So, this paragraph is trying to basically explain to us कि हमें already from ancient times only we knew what all was about inheritance and how sexual reproduction could produce various, variants of progeny. And they knew that the traits, inheritance, the heredity, was somewhere hidden in the process of reproduction. But then, and also they would put this to use very actively. They would breed selectively. They would do artificial hybridization of plants, also plants get hybrids for better breeds and stuff. So all of this happened. This shows that they did have a practical idea, but they had little knowledge scientifically on the same. Mendel's law of inheritance. Of course, whenever we talk in terms of inheritance, we have to talk about Mendel. It was during the mid 19th century that headway was made in the understanding of inheritance. Gregor Mendel conducted hybridization experiments on garden peas for 7 years. Just so you know, this date has been asked in your NEET exam. Find this. So, for 7 years, from 1856 to 1863, Mendel was working and proposed the laws of inheritance in living organisms. During Mendel's investigations into inheritance patterns, it was for the first time that statistical analysis and mathematical logic were applied to problems in biology. And this was actually also a reason as to why Mendel's laws were not widely accepted, because they thought that mathematics into biology was not a good match. His experiments had a large sampling size, which gave greater credibility to the data that he collected. Also, the confirmation of his inferences from experiments on successive generations of his test plants proved that his results pointed to general rules of inheritance rather than being unsubstantiated ideas. Mendel investigated characters in the garden pea, fine, Pisum sativum plant that were manifested as two opposing traits, example, tall or dwarf, yellow or green seeds. This allowed him to set up a basic framework of rules governing inheritance, which was expanded on by later scientists to account for all the diverse natural observations and the complexity inherent in them. Now, this is basically, NCRT would try to confuse you with the complex English language, but this is nothing to get scared of. Basically, they're trying to explain what all did Mendel do, to begin with. it's just the intro. Find how he collected the samples and how he had lots and lots of experimental data. He would collect plants with contrasting traits, and he basically took 7 pairs of contrasting characters in pea plant, and he's actually observed कि कुछ हाइब्रिडाइज करा रहे हैं प्लांट्स को, तो हमारे पास क्या रिकॉम्बिनेंट्स आ रहे हैं, व्हाट इज द रेशियोज, एंड actually bringing mathematics into biology. Fine. Mendel conducted such artificial pollination or cross pollination experiments using several true breeding pea lines. True breeding means जो आप constantly उसको homozygosity ला रहे हैं, आप constantly उसको selfing कर रहे हैं, you will eventually get a homozygous breed. A true breeding line is one that having undergone continuous self pollination shows a stable trait inheritance and expression for several generations. True breeding pea plant varieties as pairs. Fine, so 7 pairs were taken by Mendel. ठीक है, 7 किस्म के हमने variants ले लिए जिनमें different different properties थे, which was similar except for one character with contrasting traits. Fine, so in pairs he would take 7 pairs उसने लिए, and every pair would be similar, just one trait would be different. Fine, they would differ in only one character, having different traits of the same character. Some of the contrasting traits selected were smooth or wrinkled seeds, yellow or green seeds, inflated or constricted or yellow pods, and tall or dwarf plants. Now, let me tell you these properties, the contrasting traits that were taken by Mendel, they've been asked so many times you can't even imagine. NEET वाले, they really are fond of these types of questions. कौन से traits थे जो मेंडल ने लिए थे, कौन से थे जो नहीं लिए थे? So you have seed shape, you have seed color, you have flower color, you have pod shape, and all of the rest of these. Pod color hai or flower position hai, whether it's axial, it's terminal, you have stem height, whether it's a tall or a dwarf plant. Fine. All right. It's also given in a really nice tabular form. You can see stem height mein contrasting traits. Basically, what is a trait? It's the variations of the same character. What are its different types? Fine. Then you have flower color, violet or white, flower position, actual terminal, pod shape, inflated, constricted, pod color, green, yellow, seed shape, round, wrinkle, seed color, yellow, green, etc. We'll do this also in the experiments as well. Now, inheritance of one gene, when you talk in terms of a single gene, fine, तो हमारे पास monohybrid crosses आते हैं. We'll just see it right now. Let us take the example of one such hybridization experiment carried out by Mendel, where he crossed tall and dwarf pea plants to study the inheritance of one gene. अब उसने क्या लिया? Height का gene ले लिया, height का inheritance देखा. So he just took pea plants which were differing in their heights. He collected the seeds produced as a result of this cross and grew them to generate plants of the first hybrid generation. This generation is also called the filial 1 progeny or the first filial F1 generation. Mendel observed that all the F1 progeny plants were tall, like one of its parents. None were dwarf. He made similar observations for the other pairs of traits. He found that F1 always resembled either one of the parents and that the trait of the other parent was not seen in them. Again, उसने प्लांट लिया. ठीक है, अभी सबसे पहले उसने सारे कैरेक्टर्स में सेपरेटली किया, बट दे हैव गिवन सिंप्लिसिटी के लिए हम कन्वेंशनली भी हमेशा हाइट का ही ट्रेट लेते हैं. सबसे पहले हमने ले लिया, दिस इज जस्ट शोइंग कि उसने क्या किया, इमैस्कुलेशन जस्ट फॉर द पर्पस ऑफ हाइब्रिडाइजेशन, आर्टिफिशियली पॉलिनेटिंग देम. फाइन, तो इमैस्कुलेशन किया, बैगिंग, रीबैगिंग, यू हैव ऑलरेडी डन दिस बिफोर. ठीक, एंड व्हाट ही फाउंड वाज व्हेन ही सेल्फ दीज, एक उसने टॉल ले लिया, दूसरा उसने ड्वार्फ ले लिया. ट्रू ब्रीडिंग लाइन, सो ट्रू ब्रीडिंग होमोजाइगस, और जो हमारे होते हैं कैपिटल डी कैपिटल टी, स्मॉल टी, टॉल के दोनों सेम होमोजाइगस ट्रेट्स ले लिए, ड्वार्फ के सिमिलर ले लिया. दैट्स हाउ यू गेट अ ट्रू ब्रीडिंग लाइन. एंड व्हेन ही क्रॉस देम, जो उनकी प्रोजेनी थी उसमें सारे टॉल थे, एंड जो ड्वार्फ वाला था इट रिमेन्ड अन. Mendel then self-pollinated the tall F1 plants. अब ये सारे tall है, इनको self किया, and to his surprise found that in the filial second generation some of the offspring were dwarf. ठीक, अब dwarfness दूसरी generation में आ गया. The character that was not seen in the F1 generation was now expressed. The proportion of plants that were dwarf were 1/4 of the F2 plants, while 3/4 of the F2 plants were tall. So basically 1:3 के रेशियो में आप देख सकते हैं, जो ड्वार्फ थे दे वर 1/4, एंड 3 out of the 4 were tall. But हमारा जो unexpressed gene था in the first filial, now it got expressed. The tall and dwarf traits were identical to their parental type and did not show any blending. That is, all the offspring were either tall or dwarf, none were of in between height. तो blending नहीं हो रहा था basically, वो parental ही था, whether it was tall or dwarf, but they looked just like their parents. So there was not nothing of intermediate. Right? Similar results were obtained with the other traits, जैसा मैंने बोला, that he studied. Only one of the parental traits was expressed in F1, or while at the F2 stage, both traits were expressed in the proportion 3 is to 1. The contrasting traits did not show any blending at either F1 or F2. This is basically the crux of what all Mendel did. Fine, मोनोहाइब्रिड क्रॉस में यही हुआ कि पहली जनरेशन की उसमें सिर्फ एक ही ट्रेट एक्सप्रेस हुआ, दूसरी जनरेशन में दोनों ट्रेट्स एक्सप्रेस हुए इन द रेशियो 3:1, and there was no blending, fine, at either of the stage. Moving on. Based on these observations, Mendel proposed that something was being stably passed down unchanged from parent to offspring through the gametes. Obviously, हम ये भी we already know it, that what is it through which all of this gets transferred. Yeah, heredity and inheritance, it's somewhere down the line it relates to the gametes, because we're talking in terms of sexual reproduction. Fine. Over successive generations, he called these things as factors. Now we call them as genes. So Mendel didn't say कि जीन जैसा कुछ है, but he said कुछ फैक्टर है जो गमीट्स के थ्रू पेरेंट्स से ऑफस्प्रिंग में आ रहा है. Genes therefore are the units of inheritance. Fine. All right, very important again. The unit of inheritance is gene. They contain the information that is required to express a particular trait in an organism. Genes, which code for a pair of contrasting traits, are known as alleles. Allele is nothing but it's a gene. It's a form of a gene. Fine. And usually, generally, हम देखते हैं एक जीन के दो alleles होते हैं, दो contrasting traits होते हैं एक character के. They're slightly different forms of the same gene. If we use alphabetical symbols for each gene, then the capital letter used for the trait expressed at the F1 stage. Now we know that's the dominant trait that will get expressed at the F1 stage. अभी इन्हें कहा नहीं है कि dominance या कुछ, but they say जो express हो रहा है उसको हम क्या कहते हैं? Capital से denote करते हैं, small alphabet for the other trait. For example, in case of the character of height, capital T is used for the tall trait, small t for the dwarf one. They are alleles of each other because they are controlling the same character and they're contrasting pairs of uh, you know genes, but they're again form of genes only. Hence, in plants, the pair of alleles for height would be capital T, capital T, capital T, small t, or small t, small t. These are all the possibilities. Fine. Mendel also proposed that in a true breeding tall or dwarf pea variety, the allelic pair of genes for height are identical or homozygous. True breeding, I already told you there's homozygosity. You have selfed it so many times that it'll represent a certain trait only now, and that too homozygous condition में. So this is true breeding. These are called the genotype of the plant, the genetic makeup कि alleles किस चीज के बने हुए हैं. Fine. While the descriptive terms tall and dwarf are the phenotype जो express हो रहा है जो आप morphologically देख पा रहे हैं, which is visible, that is the phenotype. As Mendel found the phenotype of the F1 heterozygotic to be exactly like the TT parent in a parents, है ना? यही dominant तो capital T है, express यही हो रहा है, although genetic makeover different था, मगर phenotypically a pair tall ही हुआ. He proposed that in a pair of dissimilar factors, one dominates the others, as in the F1, and hence is called the dominant factor, while the other factor is recessive. अगर pair है dissimilar factors का, उसमें एक dominant होगा, एक recessive होगा. In this case, capital T for tallness is dominant over small t, which is for dwarfness, that is recessive. He observed identical behavior for all the other characters, trait pairs that he studied. It is convenient and logical to use the capital and lower case of an alphabetical symbol to remember this concept of dominance and recessiveness. मतलब जो dominant वो capital, recessive तो small. Fine. So it's kind of easy. Alleles can be similar as in the case of homozygotes. Homozygotes में alleles similar भी हो सकते हैं. It's not necessary they're always contrasting. And they can be also dissimilar as in case of heterozygote. और heterozygotes में ही हमारे पास dominance आता है. Since the TT plant is heterozygous for genes controlling one character, it is monohybrid, and the cross is a monohybrid cross. हम एक character, we are just talking in terms of height. Fine, so we are only considering one character which is different in the two plants and we're crossing them, observing the same traits. So this will be called as a monohybrid cross. From the observation that the recessive parental trait is expressed without any blending in the F2, we can infer that when pollen dwarfs produce gametes by the process of meiosis, the alleles of the parental pairs separate or segregate from each other and only one allele is transmitted to the gamete. होता है कि हम देखते हैं उसमें जब भी हमारा डिप्लॉयड ऑर्गेनिज्म है उसमें गमीटोजेनेसिस के लिए मियोसिस होगा और उसमें क्या होगा गमीट में ओनली वन सेट ऑफ क्रोमोसोम्स विल बी नाउ प्रेजेंट फाइन तो क्या होगा एक जीन का एक ही एलील विल बी ट्रांसमिटेड टू अ गमीट फाइन so that is how only one allele will be present over there this segregation of alleles is a random process there's a 50% chance of a gamete getting either allele as has been verified by the result process कुछ भी आ सकता है either of the two alleles this way the gamete tall plants the allele this T and the gametes of the dwarf ones have these T have the allele small t. ड्यूरिंग फर्टिलाइजेशन अब क्या हो रहा है ये दिखा रहे हैं गमीटोजेनेसिस में अगर इसके गमीट्स बनेंगे उनमें हमेशा कैपिटल टी होगा इसके बनेंगे तो हमेशा स्मॉल टी ही बनेगा ड्यूरिंग फर्टिलाइजेशन two alleles capital T from one parent through the pollen and small t from the other parent through the egg so it's just saying for example हमने एक पोलन ले लिया एक एग ले लिया not necessarily हमेशा ऐसे होगा are united to produce zygotes that have one capital T allele and one small t allele zygote में दोनों थे. In other words, the hybrids have this. Since these hybrids contain alleles which express contrasting traits, the plants are heterozygous. Fine. The production of gametes by the parents, the formation of the zygotes, F1 and F2 plants can be understood from a diagram called the Punnett square method. ये है, it was developed by British Reginald C. Punnett. It's a graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross. तो हम basically Punnett square method use करते हैं सारे genotypes निकालने के लिए and it's a really really convenient method. यहाँ पे diagrammatically they have shown a cross, monohybrid cross. एक trait में एक character में differ करे. You taking tall, you taking dwarf. ठीक? हमने माना एक मेल प्लांट था एक फीमेल प्लांट था इसने गमीट्स दिए ऑलवेज टी टी कैपिटल वंस इसने हमेशा दिया स्मॉल टी ठीक क्योंकि दोनों में से एक ही अलील आने का चांस था बट दोनों सिमिलर है होमोजाइगस दिस इज द F1 में आया हम क्या करते हैं बेसिकली दिस इज कैपिटल टी यहाँ पे हमने स्मॉल टी ले लिया इनको क्रॉस करेंगे आपस में इसको इसके साथ फिर इसको इसके साथ और इसको इसके साथ ऑल द पॉसिबिलिटीज विल बी एक्सप्लोर्ड इन द पनेट स्क्वायर जो भी हमें निकलेंगे दैट विल बी ऑल द पॉसिबिलिटीज ऑफ द जीनोटाइप्स अगेन सेल्फिंग दीज यहाँ से क्या निकलेगा कौन कौन से यूल ऑलवेज फॉर्म गमीट्स फॉर पनेट स्क्वायर मेथड कैपिटल टी स्मॉल टी तो क्या मिलेगा फिनोटिपिक रेशियो F2 का 3:1 टॉल जिसमें भी आपने डोमिनेंट टी देखा वो हमेशा टॉल ही होगा इजंट इट क्योंकि डोमिनेंट होता है एक रिसेसिव होता है और जीनोटाइप 1:2:1 fine a Punnett square used to understand typical monohybrid cross conducted by Mendel moving on we have all possible combinations possible gametes are written on upper and left column possible combinations are represented in boxes below in the squares, which generates a square output form. Punnett square shows the parental tall, male and dwarf, female plants. The male produced by them will do this progeny. हमने diagrammatically already समझा. F1 plants of genotype this are self-pollinated. The symbols this and this are used to denote the female and male respectively. The F1 plant of the genotype T small t when self-pollinated produces gametes of the genotype capital T small t. इसमें दोनों possibilities है gametes के है ना in equal proportion. When fertilization takes place, the pollen grains of genotype capital T have a 50% chance to pollinate eggs of the genotype capital T as well as स्मॉल टी तो हमेशा इसमें प्रोबेबिलिटी जो होता है 1:1 के रेशियो में होता है फाइन यू देयर इज नो सर्टेनिटी के सर्टेन एग विल गेट पॉलिनेटेड विद अ सर्टेन पोलन ग्रेन जिसका कोई सर्टेन ये होगा जीनोटाइप होगा और फिर एक पर्टिकुलर फिनोटाइप नो इट्स जस्ट इट्स अ चांस फाइन as a result of random fertilization resultant zygotes can be of the genes इसीलिए हम सारी possibilities देखते हैं कि क्या क्या possible है from the Punnett square it's easily seen that 1/4 of the random fertilizations lead to this 1/2 दो चार में से एक कैपिटल टी दो जो है हमारे हेटेरोजाइगस टॉल थे और एक जो है हमारा अगेन होमोजाइगस ड्वार्फ था फाइन externally it is not possible to distinguish between the plants with genotypes this isn't it externally दोनों क्या दिखेंगे tall दिखेंगे दोनों में T capital है dominant hence within the genotypic pair only one character tall is expressed hence the character T or tall is said to dominate over the other allele small t or dwarf character it is thus due to this dominance of one character over the other यहाँ पे हम dominance देख रहे हैं but this is allelic isn't it this is allelic dominance दो alleles में से एक डोमिनेंट होगा फॉर द सेम जीन आगे हम कुछ और भी देखेंगे एपिस्टासिस वगैरह other than other that all the F1 are tall fine though the genotype is capital T small t and in the F2 3/4 of the plants are tall though genotypically 1/2 are capital T small t and only 1/4 are capital T this leads to a phenotypic ratio of 3/4 tall and 1/4 dwarf fine दैट इज अ 3:1 रेशियो। ये यहाँ पे बता रहे हैं हमें जीनोटिपिक रेशियो। दिस इज द phenotype एंड दिस इज द genotype। फाइन, ये हमने ऑलरेडी ऊपर देखा हुआ है। दिस इज mathematically condensable to the form of binomial expression that has the gametes bearing this in equal frequency. The expression is expanded as given below। इनने यहाँ पे थोड़ा mathematics लगाया है, binomial expression किया है। Mendel self-pollinated the F2 plants and found that dwarf F2 plants continue to generate dwarf plants in F3 and F4 generations. He concluded that the genotype of the dwarf was homozygous. है ना? What do you think he would have got when he self-pollinated a tall F2 plant? अब यहाँ पे टेस्ट क्रॉस की बात आ रही है। टेस्ट क्रॉस में हम क्या करें? अभी आपने देखा, अगर हमारे पास कुछ ऐसा हो या हमारे पास कुछ ऐसा हो, इन दोनों का फिनोटाइप टॉल ही होने वाला है। अगर हमें जीनोटाइप पता करना है तो हम टेस्ट क्रॉस करते हैं, ठीक है, इसलिए इसको टेस्ट बोलते हैं। From the preceding paragraphs, it's clear that though the genotypic ratios can be calculated using mathematical probability by simply looking at the phenotype of a dominant trait, it is not possible to know the genotypic compositions. Obviously, देख के कैसे पता चलेगा genetic makeup क्या है? That is, for example, whether a tall plant from F1 or F2 has capital T or capital T small t composition cannot be predicted. जो मैंने यहाँ लिखा. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from F2 with a dwarf one. This is called a test cross. ठीक? In a typical test cross, an organism P plants here showing a dominant phenotype and whose genotype is to be determined is crossed with the recessive parent instead of self crossing। अब इसका self नहीं करेंगे, जो भी हमें मिला हम उसको क्या करेंगे, will cross it with its recessive parent which we know is homozygous dwarf। The progenies of such a cross can easily be analyzed to predict the type of test organism। We'll see it diagrammatically over here। This is the test cross conducted by Mendel। homozygous recessive है, सबसे पहले अच्छा you have a dominant phenotype, we don't know the genotype, तो genotype unknown इनने यहाँ पे लिखा हुआ है खुद ही। cool। यहाँ पे now अब इसका क्या हो सकता है, इधर इट्स डोमिनेंट दोनों, या तो एक डोमिनेंट एक रिसेसिव। हमने इसको in the both the cases we have crossed it with the homozygous recessive parental plant। यहाँ पे पनेट स्क्वायर करेंगे, इट कैन बी दिस पॉसिबिलिटी, ये भी है। यहाँ पे सारे फ्लावर्स, अच्छा they've taken in terms of color, हम हाइट भी ले सकते हैं। ऑल फ्लावर्स आर वायलेट, फिर हमारा 1 जो भी 1:1 निकला जीनोटिपिक रेशियो भी और फिनोटिपिक भी, तो यहाँ पे वी कैन से दैट इट वाज होमोजाइगस डोमिनेंट। ठीक। नाउ इफ वी हैव हाफ ऑफ द फ्लावर्स एक चीज, दूसरे हाफ वो द रिसेसिव ट्रेट, तब हमारा जो अननोन फ्लावर है दैट वाज हेटेरोजाइगस। अगेन यहाँ पे भी द रेशियो कम्स आउट टू बी 1:1। Based on his observations on monohybrid cross, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called the principles of laws of inheritance, the first law of dominance and the second law of segregation. Law of dominance. अभी हमने ऑलरेडी पढ़ा हुआ है कि दो अगर हमारे पास कॉन्ट्रास्टिंग पेयर्स ऑफ एलील्स है, उनमें से एक डोमिनेंट होगा एक रिसेसिव होगा। सिमिलर थिंग इज बीन रिटन इन पैराग्राफ्स नाउ। कैरेक्टर्स आर कंट्रोल्ड बाय डिस्क्रीट यूनिट्स कॉल्ड फैक्टर्स। उसने जीन नहीं बोला, उसने बोला फैक्टर है, तो जीन के थ्रू ट्रांसफर हो रहा है पेरेंट से प्रोजेनी में। फैक्टर्स ऑकर इन पेयर्स। ठीक, हमने देखा अभी जीन्स के दो एलील्स होते हैं। इन अ डिसिमिलर पेयर ऑफ फैक्टर्स वन मेंबर, अब यहाँ फैक्टर से जीन का मतलब है, ठीक, वन मेंबर ऑफ द पेयर डोमिनेट्स द अदर, यानी एक डोमिनेंट है दूसरा रिसेसिव है। द लॉ ऑफ डोमिनेंस is used to explain the expression of only one of the parental characters in a monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion of 3:1 obtained at the F2. What is now the law of segregation? This law is based on the fact that the alleles do not show any blending and that both the characters are recovered as such in the F2 generation, though one of these is not seen at the F1 stage. But जब ये F2 में आता है तो ये directly as its parental type आता है, it is not with any sorts of blending, it will be exactly as of its parent. Though the parents contain two alleles during gametic formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors। दोनों तो आ नहीं सकते gamete में, उसमें एक ही आएगा क्योंकि meiosis के through gametogenesis होता है। Of course, a homozygous parent produces all gametes that are similar क्योंकि उसमें choice ही एक ही है, while a heterozygous one produces two kinds of gametes। जैसे males में, in case of humans, you can have X, you can have Y। See, in females, it's always X, each having one allele with equal proportion. Now, this was about the law of dominance and law of segregation. This law of segregation or law of purity of gametes, wherein you see that without any blending, these gametes are going to, the alleles are going to separate in case of the gametes in two different gametes with an equal chance. This is a universal law and it will be followed everywhere. Fine, this is not universal. We'll see cases wherein the law of dominance will not hold true. जैसे अभी यहीं पे स्टार्ट हो रहा है हमारा incomplete dominance. Fine. Incomplete dominance क्या है? मतलब dominance नहीं हो रहा है पूरी तरीके से. When experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two. अब हम ऐसा phenotype देख रहे हैं offspring में जो parents के दरमियानी है. There's blending. It's not exactly like that of the parent. Fine. The inheritance of flower color in the dog flower, very important example, snapdragon or anti-rhinum species, a good example to understand incomplete dominance. In a true all right, in a cross between true breeding red flowers, capital R, true breeding white flowers, small r. The F1 जो हमारा ऐसे निकलेगा heterozygote was pink. ना रेड है ना व्हाइट है. हमने क्या देखा? अकॉर्डिंग टू मेंडल अगर लॉ ऑफ डोमिनेंस ही फॉलो करेंगे तो सारे F1 प्रोजेनी रेड होने चाहिए और फिर F2 में हमारे पास 3:1 के रेशियो में 3 हमारे रेड होने चाहिए, एक हमारा व्हाइट होना चाहिए. ऐसा कुछ नहीं हो रहा. When the F1 was self-pollinated, the F2 resulted in the following ratio: 1:2:1. एक red था, दो pink थे, एक white था. Here, the genotype ratios were exactly as we would expect in any Mendelian monohybrid cross, but the phenotypic ratios had changed from the 3:1 dominant is to recessive ratio. What happened was that R was not completely dominant over small r. यानी R पूरी तरीके से dominance दिखा ही नहीं पाया and made it possible to distinguish R small r as pink from capital R red and small r white. There was no complete dominance. अब क्या था concept इसके पीछे? What exactly is dominant? Why are some alleles dominant and other recessive? To tackle these questions, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular. हाँ, in a diploid organism there are two copies of each gene, that is as a pair of alleles. ये हमें पता है हर एक जीन के दो कॉपीज है और वो as alleles हमारे आगे होते हैं. Now these two alleles need not always be identical as in heterozygote. And heterozygotes में different होते हैं. One of them may be different due to some changes that it has undergone, which modifies the information that particular allele contains. Maybe during modifications, maybe during evolution, maybe as a result of mutations, this allele might have changed. अब heterozygote में alleles different है. Let's take an example of a gene that contains the information for producing an enzyme. For example, now there are two copies of this gene, the two allelic forms. हर एक जीन के दो कॉपीज है, वो दो उसके alleles है. Let us assume, as is more common, that the normal allele produces the normal enzyme that is needed for the transformation of a substrate S. Theoretically, the modified allele could be responsible for the production of the normal or less efficient enzyme, a non-functional enzyme, or no enzyme at all. अब ये ले रहे हैं basically an example कि हमारे पास एक जीन है वो एक एंजाइम प्रोड्यूस कर सकता है. अब इसके दो कॉपीज है जीन के, दो अलील्स है. ठीक. अब नॉर्मली तो हमारा अलील क्या कर रहा है, वो प्रोड्यूस कर रहा है नॉर्मल एंजाइम, लेकिन कभी कभार अलील मॉडिफाई भी हो सकता है, तो वो या तो लेस एफिशिएंट एंजाइम या नॉन फंक्शनल एंजाइम या कोई भी एंजाइम नहीं देगा. फाइन, देयर आर पॉसिबिलिटीज. In the first case, the modified allele is equivalent to the unmodified allele, that is, it will produce the same phenotype or trait, result in the transformation of substrate. Such equivalent allele pairs are very common. यानी normally था, पहले एंजाइम होता था, मॉडिफिकेशन के बाद भी ये वही कर रहा है. If the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. अब enzyme नहीं produce हो रहा है तो phenotypically we can actually observe it. The phenotype or trait will only be dependent on the functioning of the unmodified allele. Unmodified or functional allele, which represents the original phenotype, is the dominant allele, and the modified allele is generally the recessive allele. So basically how it works, अगर modification हुआ जो original था, that was a dominant one. जो modify हुआ, that becomes the recessive one. And this is how in a pair of alleles you got one dominant and the other as the recessive one. Hence, in the example above, the recessive trait is seen due to non-functional or because no enzyme is produced. ठीक, तो मॉडिफिकेशन से ऐसा हुआ कि हमने कहा कि भाई एक डोमिनेंट है दूसरा रिसेसिव है. व्हाट इज को डोमिनेंस? इनकम्प्लीट डोमिनेंस देख लिया, स्नैपड्रैगन, एंटी राइनो. Till now, we were discussing crosses where the F1 resembled either of the two parents, dominant, or was in between, incomplete dominance. But in the case of codominance, the F1 generation resembles both parents. ठीक, यानी अब दोनों पेरेंट्स से डिफरेंट नहीं बल्कि दोनों पेरेंट्स को रिजेंबल करेगा हमारा F1. ठीक, that is codominance. A good example is different types of red blood cells that determine ABO blood grouping in human beings. ABO blood groups are controlled by the gene I. ये जीन है, इसके हमारे पास तीन अलील्स है, मल्टीपल अलीलिज्म. हाँ, the plasma membrane of the red blood cells has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. The gene I has three alleles, IA, IB, and small I. Small I को IO भी लिखते हैं. The alleles IA and IB produce a slightly different form of the sugar, while allele I does not produce any sugar. Because humans are diploid, each person possesses any two of the three I genes. Obviously, we're diploid. हमें तीन में से दो ही होंगे. Fine, दो ही alleles होंगे. IA and IB are completely dominant over small I. In other words, when IA and small I are present, IA will get expressed. यानी IA और IB अगर individually small I के साथ present है because वो sugar produce कर रहे हैं तो वो dominant है. Small I does not produce any sugar. जैसे अभी हमने enzyme वाला देखा था, fine. And when IB and small I are present, IB will express because they are dominant. अब ऐसा भी possibility है तीन में से I और IB साथ में आ गए. They both express their own types of sugars and this is because of codominance. dominance. कोई छुप नहीं रहा, कोई मास्क नहीं हो रहा दूसरे के प्रेजेंस में, बट दोनों अपना एक्सप्रेशन दिखा रहे हैं. Hence, red blood cells have both A and B types of sugar. Since there are three different alleles, there are six different combinations of three alleles that are possible. Therefore, a total of six different genotypes of the human ABO blood group. क्योंकि तीन है तो उनको हम छह डिफरेंट दो दो करके छह डिफरेंट जीनोटाइप्स में सेग्रीगेट कर सकते हैं. ठीक है, तो एक पेरेंट से कुछ लिया, दूसरे पेरेंट से कुछ लिया, फिर ऑफस्प्रिंग का कुछ एक जीनोटाइप आया एंड यू हैड द ब्लड टाइप. दिस इज द phenotype दैट यू कुड एक्चुअली सी. तो अगर आए आए सिमिलरली सिंपली आए ही आएगा. ए बी साथ में है तो ए बी दोनों आएंगे. दिस इज को डोमिनेंस, दोनों एक्सप्रेस कर रहे हैं. स्मॉल आई के साथ स्मॉल आई तो है ही रिसेसिव, उसके साथ किसी को भी डालो वो डोमिनेंट बन जाता है. फाइन. और अगर दोनों स्मॉल आई है देन इट इज ओ ब्लड ग्रुप. फाइन. अब आपने ऐसे भी सुना होगा about AB positive, negative. That's about the rhesus factor. That's something else. You don't have to discuss, but that's another. You can say another. That's completely goes parallel to the normal blood groups that we are having here. Do you realize the example of ABO blood grouping also provides a good example of multiple alleles? Here you can see that there are more than two, that is three alleles governing the same character. Blood group के लिए तीन alleles. Since in an individual only two alleles can be present, multiple alleles can be found only when population studies are made. So in a group of organisms you can say कि तीन alleles possible है, but एक organism में दो ही होंगे. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. कभी कभार एक जीन का एक ही इफेक्ट होगा. It has two alleles, capital B, small b. Starch is synthesized effectively by capital B homozygotes and therefore large starch grains are produced. In contrast, ये हमारे recessive है. They have lesser efficiency in starch synthesis and produce smaller starch grains. After maturation of the seeds, these seeds are round and these are wrinkled. ये सिमिलरली आपको सिंपली दिखा रहा है डोमिनेंट कौन था, रिसेसिव कौन था, स्टार्च ज्यादा काम प्रोड्यूस होगा, कम काम प्रोड्यूस होगा. हेटेरोजाइगोट्स प्रोड्यूस राउंड सीड्स and so B seems to be the dominant allele, but the starch grains produced are of intermediate size, capital B small b seeds. So if starch grain size is considered as the phenotype, that from this angle the allele shows incomplete dominance क्योंकि intermediate height है, तो इसको भी आप starch grain का जो height है उसको आप incomplete dominance कह सकते हैं. Therefore dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much on the gene product and the production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine. In case more than one phenotype is influenced by the same gene, it happens many a times a single gene would control many different phenotypes. Inheritance of two genes. अब हमारे dihybrid cross पे, ठीक. Mendel also worked with and crossed pea plants that differed in two characters, as is seen in the cross between a pea plant that has seeds with yellow color, round shape, one that had seeds of green color and wrinkled shape. Mendel found that the seeds resulting from the crossing of the parents had yellow colored and round shaped seeds. ठीक, यानी इसका मतलब जब F1 आया, F1 में जो एक्सप्रेस होता है वही डोमिनेंट है, यानी येलो कलर इज डोमिनेंट एंड राउंड शेप इज डोमिनेंट. Thus, yellow color was dominant over green, round shape dominant over wrinkle. F1 से ही पता चलता है. These results were identical to those that he got when he made separate monohybrid crosses between yellow and green seeded plants. So monohybrid से different था. Let us use the genotypic symbols Y for dominant yellow seed color and small y for recessive green seed color, capital R for round shaped seeds and small r for wrinkled seed shape. The genotype of the parents can then be written as capital R, capital R, small y, capital Y, and this. It's really difficult to say this actually, but I just hope you're here with me with the pen in your hand and your book in front. So then it would be a lot more easier. I just cannot do, things. They're written though, but still you just cannot do things just by reading the crosses and everything. You got to do that. Do this. Fine. The cross between the two plants can be written down as in figure. Again, there's something diagrammatic given. I just hope it's said. Yeah. So I think this will do wonders for us. This is the parental generation, P generation. You have round yellow, this is dominant दोनों. You have wrinkled green, again दोनों recessive. Gametes जो produce होंगे उनमें इसका एक, इस कैरेक्टर का एक आएगा, इस कैरेक्टर का एक आएगा. अब यहाँ दोनों सेम सेम ही आएंगे, राइट? तो एक यहाँ से और इसका इस वाले कैरेक्टर का और इस वाले कैरेक्टर का. फाइन. नाउ गमीट्स आ गए. नाउ विल हैव टू फाइंड आउट. ये हमारा F1 हाइब्रिड बन गया. फाइन, यू विल सेल्फ इट. इसके बाद इसमें क्या होगा, हम लेंगे एक एक. इसको जब सेल्फ करेंगे इसके गमीट्स निकलेंगे ना सेपरेटली. ठीक, तो इसके दो कैरेक्टर्स निकलेंगे यहाँ से, इसके और इसके दो, इस कैरेक्टर के दो और इस कैरेक्टर के दो. तो हमें क्योंकि दोनों कैरेक्टर्स कंसिडर करते हैं, इसके एक इसके साथ, then इसकी इसके साथ, इसका इसका. फाइन, यू विल गेट 4 डिफरेंट कॉम्बिनेशन. तो वही फीमेल मेल गमीट्स दिया हुआ है यहाँ पे. फाइन, एंड यू कैन सी ओवर हियर, इनको क्रॉस किया है बाय सिंपली पनेट स्क्वायर. ठीक है, मल्टीप्लाई किया है, साथ में जोड़ा है इनको, और हमें मिल जाते हैं हमारे. इन they've shown it beautifully even with in the diagram they've shown कि हमारे पास राउंड येलो रिंकल्ड ग्रीन का क्रॉस हुआ है. And this is the phenotypic ratio. It's better you remember these ratios only. Fine, you will probably not get time to actually solve the Punnett square while you're in your exams. It's 9 is to 3 is to 3 is to 1. Fine. Moving on, we have law of independent assortment. All right, let's get started. In the dihybrid cross, the phenotypes round yellow, round green, and wrinkled green appeared in the ratio this. Such a ratio was observed for several pairs of characters that Mendel studied. Fine. तो राउंड येलो पहले लिया, उसके बाद हमने लिया wrinkled येलो, फिर राउंड ग्रीन और wrinkled green. The ratio of 9 is to 3 is to 3 is to 1 can be derived as a combination series of 3 yellow, 1 green with 3 round is to 1 wrinkled. This derivation can be written as follows. Again, based upon such observations on dihybrid crosses, Mendel proposed a second set of generalizations that we call Mendel's law of independent assortment. The law states that when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters. यानी दो characters के बीच में blending नहीं होगा. I think that's how even Mendel was able to perform the experiment. दो different traits लेके वो आपस में they'll be independent of each other. एक trait height का trait independent of color का trait. Fine, वो independently they'll be inherited. तो उनमें कोई भी issue नहीं है. Obviously it would have been a mess otherwise. अगर characters ही mix हो रहे हैं तो the color character would get mixed with the your height character and it would be really messy. The Punnett square can be effectively used to understand the independent segregation of the two pairs of genes. Again, in यहाँ पे Punnett square use किया. It's all this entire paragraph is about the diagram that I've already shown. Now, besides each gamete having either capital R, small I, it should have also have the allele capital Y or small y. So again, we have over here, you can easily understand that हमारे पास इस पूरे पैराग्राफ का एक क्रक्स है which which you can easily understand कि हमारे पास अगर दो डिफरेंट कैरेक्टर्स हमने लिए, वो डिफरेंट अलील्स जो है वो डिफरेंट गमीट्स जो दे रहे हैं. ठीक है, डिफरेंट गमीट्स उनमें डिफरेंट डिफरेंट. अब हमने दो कैरेक्टर्स लिए तो हम चार अलील्स कंसिडर करेंगे. अब जो इंडिपेंडेंटली हर एक कैरेक्टर के दो दो अलील्स हैं उनका सेग्रीगेशन जो है इट इज इंडिपेंडेंट ऑफ द अदर पेयर. ठीक है वो. और ऑल्सो you will have a possibility of four gametes. That's what it's trying to say. और उनमें with a frequency of 25%. और ये हमारे पास यहाँ पे four genotypes of gametes because four types of pollens and four types of eggs will be produced. We are considering two characters, so दो दो होंगे हर एक में. Fine. वही इसने यहाँ पे कहा है. Chromosomal theory of inheritance, again it was given by Bowery and Sutton. Fine. Mendel published his work on inheritance of characters in 1865, but for several reasons it remained unrecognized till 1900. Firstly, the communication was not easy as it is now in those days, and his work could not be widely publicized. Okay? Secondly, his concepts of genes or factors in Mendel's words as stable and discrete units that control the expression of traits and of the pairs of alleles which did not blend with each other was not accepted by his contemporaries as an explanation for the apparently continuous variation seen in nature. Fine. So again it's trying to say here over कि जो मेंडल ने दिया था, Mendel's work why it remained hidden, or at that time it had various reasons, and many a times people were not ready to accept mathematics into biology, even that was a reason. Thirdly, Mendel's approach, exactly what it's trying to say over here, using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, though Mendel's work suggested that factors or genes were discrete units, he could not provide any physical proof for the existence of factors or say what they were made of. तो उसने बस बोला कि factors है, transmit हो रहे हैं, inherit हो रहे हैं. He didn't give exactly कि था क्या factor, fine. In 1900, we had D. Rees, Correns, T. Shermark independently rediscovered Mendel's results on the inheritance of characters. इन्होंने इन तीन scientists ने जो है they rediscovered Mendel's work and they showed इनके हर एक inheritance का जो भी इनने experiment दिया था उसको laws में put in किया. Also by this time due to advancement in microscopy that was taking place, scientists were able to carefully observe cell division. Again microscope आ गया. This led to the discovery of structures in the nucleus that appear to double and divide just before each cell division. इनने देखा कुछ ऐसी चीजें हैं जो सेल डिवीजन से बिल्कुल पहले डिवाइड हो रहे हैं, फाइन. और इनको बोला क्रोमोसोम्स, colored bodies as they were visualized by staining. Chroma means color. By 1902, the chromosome movement during meiosis had been worked out. अब वो meiosis को देख रहे हैं, उसमें chromosomes का interference देख रहे हैं. Walter Sutton and Theodore Bowry noted the behavior of chromosomes was parallel to the behavior of genes and used chromosome movement to explain Mendel's law. अभी पता नहीं है कि gene actually chromosome पर ही lie कर रहे हैं. Gene ही है जो chromosome बना रहा है. लेकिन ये लोग क्या कह रहे हैं? When Sutton and Bowry came, they said that it goes parallel. जो genes कर रहे हैं, genes divide हो रहे हैं, फिर gene segregate हो रहा है, फिर gene inherit हो रहा है, वही chromosomes का भी same हाल है. Fine. Recall that you've studied the behavior of chromosomes during mitosis and during meiosis. The important things to remember is that chromosomes as well as genes occur in pairs, है ना? उनने देखा chromosome भी pair पे आ रहे हैं, homologous chromosomes. Though alleles of a gene pair, again, they're located on homologous sites of homologous chromosomes because they go both in parallel. And what is the comparison? Beautiful table over here. Pehla wala is given about the chromosomes, the second one about the genes. Occurs in pairs, segregate at the time of gamete formation, such that only one of each pair is transmitted. Independent pairs segregate independently of each other. ठीक. During anaphase of meiosis 1, the two chromosome pairs can align at the metaphase plate independently of each other. To understand this, compare the chromosomes of four different colors in the left and right columns. In the left column, possibility 1, orange and green is segregating together, but in the right hand column, orange chromosome is segregating with the red chromosomes. अब यहाँ पे इन्हें दो possibilities दिया. One long orange and short green, one long orange short red chromosome yellow. What is happening? This is basically all of your cell cycle and cell division. ठीक है, meiosis 1 ले लिया है और anaphase ले लिया है. Again, two different cases are taken over here. तो ये बेसिकली हमें दिखा रहे हैं कि एनाफेज पे क्या हो रहा है. मेटाफेजिक प्लेट पे तो अलग अलग ये लोग रह गए, सेग्रीगेट रह गए, और जब ये लोग सेपरेट हो रहे हैं मियोसिस सेकेंड वाले एनाफेज में, अगेन इंडिपेंडेंट ऑफ ईच अदर यू कैन सी देम सेपरेटिंग. ऑरेंज और ग्रीन में कोई ब्लेंडिंग नहीं है, येलो रेड में कोई ब्लेंडिंग नहीं है. फाइन. सिमिलर फॉर हियर फॉर द germ cells. Again, this is just independent assortment of chromosomes, nothing to go right deep into it. Sutton and Bowry argued that the pairing and separation of a pair of chromosomes lead to the segregation of a pair of factors they carried. Sutton united the knowledge of chromosomal segregation with Mendelian principles and he called it chromosomal theory of inheritance. तो इनने बस बोला. We had somebody that is Morgan, who is also known as the father of experimental genetics, who experimentally proved chromosomal theory of inheritance. Following this synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to discovering the basis for the variation in sexual reproduction. Peak variation आ कहाँ से रहा था? Morgan worked with the tiny fruit flies Drosophila melanogaster, which were found very suitable for such studies. They could be grown on simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks, and a single male could produce a large number of progeny. So obviously, he had done the statistics, analyzed कौन सा organism मेरे लिए ठीक रहेगा. Also, there was a clear differentiation of the sex of male and female flies are easily distinguishable. Also, it many types of hereditary variations that can be seen with low power microscopes. Linkage and recombination. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex linked. The crosses were similar to the dihybrid crosses carried out by Mendel in peas. For example, Morgan hybridized yellow bodied, white eyed females to brown bodied, red eyed males and intercrossed the F1 progeny. He observed that the two genes did not segregate independently of each other and the F2 ratio was deviated very significantly from the 9 is to 3 is to 3 is to 1 ratio, which was expected when the two genes are independent. अब इसने क्या बोला? Independently कोई भी gene segregate नहीं कर रहे हैं. Fine. Morgan and his group knew that the genes were located on the X chromosome and saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combinations were much higher than the non-parental type. यानी जो parent का already combination है उसका ज़्यादा proportion आएगा, उसका ज़्यादा percentage, उसका ज़्यादा probability है progeny में आने का. Morgan attributed this due to the physical association or linkage of the two genes. हाँ, linkage वगैरह भी Morgan ने ही दिया, and coined the term linkage to describe this physical association of genes on a chromosome, and the term recombination to describe the generation of non-parental gene combinations. अब क्या हो रहा है? Recombination हो रहा है, linkage हो रहा है genes को on the same chromosome. Morgan and his groups also found that even when genes were grouped on the same chromosome, some genes were very tightly linked, जिनने बहुत कम recombination दिखाया, while others were loosely linked, which showed higher recombination. For example, he found that the genes white and yellow were very tightly linked and showed only 1.3% recombination, while white and miniature wing showed 37.2% recombination. ये दो different traits है, ये दो different genes है, yet they are showing combination, this linkage. His student Alfred Sturtevant used the frequency of recombination between gene pairs on the same chromosome as a measure of the distance between genes and mapped their position on the chromosome. What did Sturtevant say? He said that ये क्या frequency है कि recombination होगा between gene pairs on the same chromosome depends upon the distance. Today, genetic maps are extensively used as a starting point in the sequencing of whole genomes, as was done in the case of Human Genome Sequencing Project, which is discussed later. ठीक, यहाँ पे इसने बेसिकली बोला कि डिस्टेंस जो है हमारा उस पर डिपेंड करता है कि कितनी फ्रीक्वेंसी से रिकॉम्बिनेशन होगा या कितना लिंकेज होगा ऑफ द क्रोमोसोम्स ऑफ द जीन्स बेसिकली प्रेजेंट ऑन द सेम क्रोमोसोम. Polygenic inheritance. Mendel's study mainly described those traits that have distinct alternate forms, flower color, which are either purple or white. But if you look around, you'll find that there are many traits which are not so distinct in their occurrence and are spread across a gradient. For example, in humans, we don't just have tall or short as two distinct alternatives, but a whole range of possible heights, isn't it? मेंडल ने तो लिया एक बाई टॉल है एक ड्वार्फ है, ये हमारा टॉल पी प्लांट है, ये हमारा ड्वार्फ पी प्लांट है. But this does not hold true in every case. हमारे में ही आप height का range देख सकते हैं. You can just look around. There's a huge range. Such traits are generally controlled by three or more genes and are thus called polygenic traits. ऐसे traits जो बहुत variation दिखाते हैं, long range variations, and are controlled by three or more genes, उनको हम polygenic traits बोलते हैं. Besides the involvement of multiple genes, polygenic inheritance also takes into account the influence of environment. Human skin color is another example for this. In a polygenic trait, the phenotype reflects the contribution of each allele. That is, the effect of each allele is additive. ठीक, हर एक allele का जो भी action होगा वो add हो जाएगा. To understand this better, let us assume that three genes A, B, C control skin color in human with the dominant forms A, B, and C, responsible for dark skin color, recessive forms for light skin color. The genotype with all the dominant alleles will have the darkest skin color, and that with all the recessive alleles will have the lightest skin color. अब हमने तीन genes लिए, उसके छह alleles ले लिए, एक में सारे dominant लिए, दूसरे में सारे recessive लिए, for basically for your skin color. As expected, the genotype with three dominant alleles and three recessive alleles will have an intermediate skin color. In this manner, the number of each type of alleles in the genotype would determine the darkness or lightness of the skin in an individual. यानी कितने dark है, कितने light है, this will depict actually the skin color. Again, you can see a large variation over skin color. Also, pleiotropy. We have so far seen the effect of a gene on a single phenotype or trait. एक gene एक ही phenotype और character या trait को depict कर रहा था. But however, a single gene can exhibit multiple phenotypic expression. एक gene है, बहुत सारे characters को क्या कर रहा है, कंट्रोल कर रहा है. ऐसे जीन को प्लियोट्रोपिक जीन बोलते हैं. फाइन. द अंडरलाइंग मैकेनिज्म ऑफ प्लियोट्रोपी इन मोस्ट केसेस द इफेक्ट ऑफ अ जीन ऑन मेटाबॉलिक pathways contribute to different phenotypes. An example of this is the disease phenylketonuria, pleiotropy. PP you can remember, which occurs in humans. The disease is caused by mutation in the gene that codes for the enzyme phenylalanine hydroxylase. तो ये वाला enzyme जो है produce नहीं होगा, तो basically हमारा जो है phenylalanine it won't get converted into tyrosine because phenylalanine hydroxylase has not been produced. Fine. So mutation on it. This manifests itself through phenotypic expression characterized by mental retardation and reduction in hair and skin pigmentation. यानी एक ही जीन है, इससे बहुत सारे phenotype जो है वो affect हो रहे हैं. एक तो एंजाइम नहीं बनेगा, mentally भी retard होगा बंदा, और reduction in hair, skin pigmentation, all of this by a single gene. Fine, this is single gene mutation. Sex determination. The mechanism of sex determination has always been a puzzle before the geneticists. The initial clue about the genetic chromosomal mechanism of sex determination can be traced back to some of the experiments carried out in insects. तो कैसे बोलेंगे मेल है या female है? Fine, very easy. In fact, the cytological observations made in a number of insects led to the development of the concept of sex or chromosomal basis of determination. Fine, Henking could trace a specific nucleus called spermatogenesis. It was also observed by him that 50% of the sperm received the structure of spermatogenesis, whereas the other 50% did not receive it. Henking ने क्या बोला? कोई ऐसी body है. He gave this name X body. He said that there's this X body जो 50% में जा रहा है, 50% में नहीं जा रहा. He could not explain its significance. Further investigations by other scientists led to the conclusion that the X body of Henking was in fact a chromosome, and that is why it was given the name X chromosome. ठीक basically एक कौन सी body थी? It was nothing but it was a chromosome which was getting transferred. It was also observed that in a large number of insects, the mechanism of sex determination is the XO type. That is, all eggs bear an additional X chromosome besides the other chromosomes or autosomes. We have autosomes and allosomes, we know it already. तो क्या हो रहा था इंसेक्ट्स में कैसा मैकेनिज्म है एक्सओ टाइप का है सब में नॉर्मल होगा बट फीमेल्स में एक एक्स्ट्रा एक्स होने वाला है. On the other hand, some of the sperms bear the X chromosome, whereas some do not. Eggs fertilized by sperm having an X chromosome become females, and those fertilized by sperms that do not have an X chromosome become males. तो जो बेसिकली क्या होगा eggs जो है अगर X जीरो टाइप है हमारे पास फाइन तो X जीरो में क्या हो रहा है इफ egg is fertilized by a sperm having an X then it will become a female फाइन और अगर ऐसा हो रहा है कि एक्स बॉडी नहीं है हम इंसेक्ट्स में ही हैंकिंग ने देखा था अगर एक्स बॉडी ही नहीं है तो क्या बनेगा हमारे पास एक्स जीरो दैट इज देयर बी नो एक्स क्रोमोसोम दैट विल बी अ मेल fine due to the involvement of the X chromosome in the determination of sex it was designated to be the sex chromosome and the rest of the chromosomes were named as autosomes. ठीक grasshopper is an example of X0 type of sex determination in which the males have only one X chromosome besides the autosomes whereas females have a pair of X chromosomes. Fine. So basically इसमें क्या हो रहा है? Males will have X0, they'll have only one X, and females will have two X chromosomes. Clear? Next, these observations led to the investigation of a number of species to understand the mechanism of sex determination. In a number of other insects and mammals, including man, XY type of sex determination is seen where both male and female have same number of chromosomes. Same number है दोनों में. Insects में क्या होता था? Females में एक ज़्यादा होता है, isn't it? Among the males, an X chromosome is present, but its counterpart is distinctly smaller and called the Y chromosome. Females, however, have a pair of X chromosomes. Both males and females bear same number of autosomes. Hence, the males have autosomes plus XY, while the females have autosomes plus XX. In human beings and in Drosophila, the males have one X and one Y, whereas females have a pair of X chromosomes. The Drosophila may be same हो रहा है. In the above description, you've studied about two types of sex determining mechanisms, that is XO type and XX type. But in both cases, males produce two different types of gametes, either with or without an X chromosome. पहले case में some with X and some with Y. दूसरे case में ऐसे such types of sex determination mechanism is designated to be the example of male heterogamety. यानी males में दो options आ जाते हैं या तो एक में से फॉर एग्जांपल इन केस ऑफ इंसेक्ट्स एक में एक्स बॉडी आएगी दूसरे में नहीं आएगी दिस इज हेटेरोगमी in some other organisms example birds में टेढ़ा होता है different mechanism is observed in this case the total number of chromosome is same in both males and females but two different types of gametes in terms of the sex chromosomes are produced by females यानी female heterogamety where do you see this ऐसे तो females homogametic होते हैं but in case of birds you see female heterogamety fine in order to have a distinction with the mechanism of sex determination described earlier, the two different sex chromosomes of a female bird have been designated to be the Z and W chromosome because it's different. In these, so you have a ZW type. In these organisms, the females have one Z and one W chromosome, whereas males have a pair of Z chromosome besides the autosome. So males will have ZZ and females have ZW. This is female heterogamety prominent in birds. Now, in case of humans, very easy. You've been doing this 8th class se shayad. It has already been mentioned the sex determining mechanism in case of humans is XY type. Out of the 23 pairs of chromosomes, 22 are exactly same in both males and females. They are the autosomes. See, एक pair of X chromosomes present in the female, whereas the presence of an X and Y are determinant of the male characteristic. During spermatogenesis among males, two types of gametes are produced. 50% produce carry the X chromosome and the 50% has Y chromosome because दो possibilities है but equal proportion में ठीक. Besides autosomes, females however produce only one type of ovum with an X chromosome. हाँ एक ही है. There's an equal probability of fertilization of the ovum with the sperm containing either X or Y. In case the ovum fertilizes a sperm carrying X chromosome, the zygote develops into a female and the fertilization of ovum. Ovum तो static है, उसमें हमेशा X ही आएगा. Now about the sperm, it can have either X or Y. If it's X, then it's a female. Then if it's Y, carrying sperm results into a male offspring. Thus, it is evident that if genetic makes of the sperm that determines the sex of the child, it's also evident that in each progeny, uh, sorry, in each pregnancy, there's always 50% probability of either a male or a female child. It's unfortunate that in our society, women are blamed for giving birth to female children and have been ostracized and ill-treated because of this false notion. It's like a very, very old orthodox notion that it's the female, it's the mother who's responsible for the, sex of the child. But it's not so. Now you're science students and you know that it's the male sperm composition because there's a chance it can either be X or it can be Y during gametogenesis because दोनों में से एक ही आएगा. Fine. Now honeybee. Why is it so differently written over here? Let's see. The sex determination in honeybees is based on the number of sets of chromosomes an individual receives. It is said to receive. An offspring formed from the union of a sperm and an egg develops as a female, queen or a worker. Female में दो ही होते हैं, queen or a worker. And an unfertilized egg develops as a male by parthenogenesis. So what is the means? Drone कहाँ से बनते हैं? Female कहाँ से male कहाँ से बनते हैं? They are formed by parthenogenesis. यानी वहाँ fertilization नहीं होता. This means the male will have number of chromosomes than that of a female क्योंकि वो बने ही वैसे हैं. तो female haploid क्योंकि वो fertilization साथ बने हैं having 34 chromosomes males and haploid having 16 chromosomes. This is very important. Honeybee में सवाल आता है because it's really different. The mechanism is really different. So you can see 32 in a diploid female. Female can be queen or worker. दोनों ही fertilization से बनते हैं. Drones parthenogenesis से बनते हैं 16 ही होंगे. This is called as haplodiploid sex determination system and has special characteristic features such as the males produce sperms by mitosis. क्यों mitosis है? उनको meiosis करने की जरूरत ही नहीं है. वो already haploid है ना? They do not have father and thus cannot have sons but have a grandfather and can have grandsons. Is it clear? कि directly जो है क्योंकि उसको mitosis करना है वो तो meiosis से दे नहीं सकता but they can have grandsons, they can have a grandfather. How is the sex determination mechanism different in the birds? Birds में क्या होता था? Is the sperm or the egg responsible for the sex of the child? It's the opposite because heterogamety egg पे होता है, isn't it? Birds में you have female heterogamety. Now you have mutation is a phenomenon which results in alteration of DNA sequences and consequently results in changes in the genotype and the phenotype of an organism. In addition to recombination, mutation is another phenomenon that leads to variation in DNA. तो एक तो हमारा recombination से आता था alter होता था हमारा DNA और अब mutation भी है जिसकी वजह से DNA जो है वो alter हो सकता है. As you will learn, one DNA helix runs continuously from one end to the other in each chromatid in a highly supercoiled form. Therefore, loss or deletions or gain of a segment of DNA result in alteration in chromosomes. Since genes are known to be located on chromosomes, alteration in chromosome results in abnormalities or aberrations. Chromosomal aberrations are commonly observed in cancer cells. In addition to the above, mutation also arises due to change in a single base pair of DNA. This is known as point mutation. यानी एक base pair में change आ रहा है उसको point mutation करें. Classical example of such a mutation is sickle cell anemia. Sickle cell anemia में क्या हो रहा है? Point mutation. Deletions and insertions was frame shift mutation. Fine. अब हमारे पास म्यूटेशन जो है पॉइंट म्यूटेशन हो सकता है, फ्रेम शिफ्ट हो सकता है. पॉइंट म्यूटेशन मतलब बेसिकली हमारा एक सिंगल बेस पेयर चेंज हो रहा है. अगर डिलीशंस हो रहे हैं, इंसर्शंस हो रहे हैं, उसको फ्रेम शिफ्ट म्यूटेशन कहते हैं. The mechanism of mutation is beyond the scope of this discussion. At this level, there are many mutagens किनको बोलते हैं. So the mutagens are basically those physical factors, chemical factors which will induce mutation. So it can be UV radiations or anything. Let's come to genetic disorders now, wherein you'll have to do the pedigree analysis, basically looking at the history of the family history of the organism so that you can make an idea that where did the disease actually come from. Whenever we talk in terms of genetic disorders, you have to talk about pedigree analysis because they're usually inherited. Fine. The idea that disorders are inherited has been prevailing in the human society since long. This was based on the heritability of certain characteristics, features, and families. After the rediscovery of Mendel's work in the practice of analyzing inheritance pattern of traits in human beings began, since it is evident that control crosses that can be performed in pea plant or some other organisms are not possible in case of human beings, study of the family history about inheritance of a particular trait provides an alternative. Such an analysis of traits in a several of generations of a family is called the pedigree analysis. That you'll go through several generations and you'll see what is this family tree, and all of the generations, what do they depict, what diseases, what is their genetic makeover. Fine. In human genetics, pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality, or disease. Some of the important standard symbols used in the pedigree analysis are given over here. ठीक है, pedigree analysis के symbols you have male, square, female, circle, rhombus, affected individuals color डाला है, male में single line आएंगे, show mating between relatives, ठीक, parents above, children below, vertically आप children दिखाते हैं, affected disease, fine. Next, as you've studied, each and every feature in any organism is controlled by one or the other gene located on the DNA present in the chromosome. DNA is the carrier of genetic information. Hmm? It's hence transmitted from one generation to the other without any change or alteration. However, changes or alteration do take place occasionally. An alteration or change in the genetic material is referred to as mutation, which we've already done. DNA तो as it is ही आएगा, but कभी कभार उसमें क्या है, changes भी आते हैं, alterations आते हैं. A number of disorders in human beings have been found to be associated with the inheritance of changed or altered genes or chromosomes. Let's come to Mendelian disorders now. Broadly, genetic disorders may be grouped into two characters. You can categories. You can have Mendelian disorders and you can have chromosomal disorders. Mendelian disorders are mainly determined by alteration or mutation in the single gene. These disorders are transmitted to the offspring on the same lines as we have studied. The pattern of inheritance of such Mendelian disorders can be traced in a family by the pedigree analysis. Most common and prevalent Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. It is important to mention here that such Mendelian disorders may be dominant or recessive. Hmm. By pedigree analysis, one can easily understand whether the trait in question is dominant or recessive. Similarly, the trait may also be linked to the sex chromosome, as in case of hemophilia. It is evident that this X-linked recessive trait shows transmission from carrier female to male progeny. A representative pedigree is shown. You can see it over here. Fine, you're given mating, you're giving the amount of the individual's progeny, the ones which are affected, the ones which are not. Pehla wala is autosomal dominant trait. Myotonic dystrophy is autosomal dominant. You have autosomal recessive में you have sickle cell anemia. Hmm? Color blindness now in यहाँ से भी सवाल आते हैं, is a sex linked recessive disorder due to defect in either red or green cone of eye resulting in failure to discriminate between red and green color. This defect is due to mutation in certain genes present in the X chromosome. ठीक है, mutation हुआ है X chromosome में. It occurs in about 8% of males and only about 0.4% of females because the genes that lead to red green color blindness are on the X chromosome. Males have only one and females have two. Fine. The son of a woman who carries the gene has a 50% chance of being colorblind. The mother is not herself colorblind because the gene is recessive. That means that its effect is suppressed by her matching dominant normal gene. इसलिए हम इसको recessive. Daughter will not normally be colorblind unless her mother is a carrier and her father is colorblind. Hemophilia. This sex-linked recessive disease, which shows its transmission from unaffected carrier female to some of the male progeny, has been widely studied. In this disease, a single protein that's a part of the cascade of proteins involved in the clotting of blood is affected. The blood clotting will be affected. Fine. Due to this, in an affected individual, a simple cut will result in non-stop bleeding. The heterozygous female, the carrier for hemophilia, may transmit the disease to sons. The possibility of a female becoming a hemophilic is extremely rare because mother of such a female has to be at least carrier and the father should be hemophilic, which is unviable in the later stage of life. So he won't eventually get married. The family pedigree of Queen Victoria shows a number of hemophilic descendants as she was a carrier of the disease. तो अगर क्लॉटिंग नहीं हो रहा है ना, सिंपल क्लॉट कट वुड लीड टू लिटरली अनस्टॉपेबल ब्लीडिंग जिसको आप स्टॉप नहीं कर सकते कम व्हाट में, एंड इवेंचुअली द पर्सन विल डाई. तो ऐसे लोग दे रियली unviable in the later stage, like they cannot survive for long. Fine, and basically the female उसका XX होता है because this is an X linked disorder, तो उसमें दो X होते हैं, तो dominant वही होगा जो normal वाला allele होगा. Fine. तो usually it's normal, but जब males के case में they have a 50% probability कि उनका जो X होगा वो क्या होगा, that is going to be, if their mother's a carrier, and then it's very easy. Obviously जब उनकी माँ carrier होगी, then they have an XY, the Y from the male parent from father, and Y though will be normal, X from female can be, it's a 50% probability of carrying the disease. Sickle cell anemia, there's an autosome linked, इस पे बहुत सवाल आते हैं, autosomal है, X linked है, recessive है, very easy. Autosome linked recessive trait that can be transmitted from parents to the offspring when both the parents are carrier for the gene, जब दोनों ही heterozygous होंगे, यानी HBA HBS. The disease is controlled by a single pair of allele, HBA and HBS. Out of the three possible genotypes, only homozygous individuals for HBS show the diseased phenotype. अब तीन होते हैं ना हमारे पास possibility, दोनों A दोनों S या एक S एक A. अब homozygous individuals जो है they will show the diseased phenotype HBS HBS. So for this both the parents have to be carriers for the gene. Heterozygous दोनों को होना पड़ेगा. Heterozygous HBA HBS individuals appear apparently unaffected but they're carrier of the disease because there's a 50% probability. अब इनमें HBS है ना तो ये carrier बन जाएंगे हालांकि खुद ये unaffected हो of the mutant gene to the progeny, thus exhibiting sickle cell trait. The defect is caused by the substitution of glutamic acid, basically GLU, by valine. Okay, GAG GUG में convert हो रहा है, you can just remember, at the sixth position of the beta globin chain of the hemoglobin. Hemoglobin में तो alpha होते हैं तो beta होते हैं, तो sixth position में क्या हो रहा है, glutamic acid is being converted into valine. इसी की वजह से sickle cell anemia हो गया. The substitution of amino acid in the globin protein results in single base substitution of the sixth codon of the beta globin gene from GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of the RBC from biconcave disc to elongated sickle-like structure. Moving on, phenylketonuria. This inborn error of metabolism is basically error of metabolism is also inherited as the autosomal recessive. Autosomal recessive, इससे पहले ही हमने देखा क्या होता है, एंजाइम नहीं है, ठीक है, there is no enzyme which can convert phenylalanine into tyrosine. As a result of this, phenylalanine is accumulated, converted into phenylpyruvic acid and other derivatives. Accumulation of these in brain results in mental retardation. Yeah, अभी इसका I guess pleiotropy भी किया हुआ था, right? This is also excreted through urine because of its poor absorption by kidney. Coming to thalassemia, there's also an autosome linked recessive blood disease. ठीक है, autosome linked है recessive blood disease transmitted from parents to the offspring when both the partners are unaffected carriers for the gene. दोनों ही unaffected है लेकिन दोनों heterozygotes है, दोनों carriers है. The defect could be due to either mutation or deletion, which ultimately results in reduced rate of synthesis of one of the globin chains, alpha या beta, that make up hemoglobin. Blood का disorder है तो hemoglobin के जो दो चेन्स है ना अल्फा बीटा, उसका जो बनने का सिंथेसिस का जो रेट है या तो वो कम हो जाता है. This causes the formation of abnormal hemoglobin molecules resulting into anemia, which is characteristic of the disease. Anemia हो गया तो thalassemia is also possible. Thalassemia can be classified according to which chain of the hemoglobin molecule is affected. कौन सा hemoglobin chain बन नहीं रहा, ठीक है, किसके वजह से abnormal hemoglobin आ रहा है. In alpha thalassemia, production of alpha globin chain is affected, while in beta thalassemia, production of beta globin chain is affected. Very easy. Alpha thalassemia is controlled by two closely linked HBA1 and HBA2 on chromosome 16 of each parent, and it is observed due to mutation or deletion of one or more of the four genes. See? The more genes affected, the less alpha globin molecules produced, while beta thalassemia is controlled by a single gene, HBB, on chromosome 11 of each parent and occurs due to mutation of one or both the genes. More or less, याद रखना है, thalassemia differs from sickle cell anemia in that the former is a quantitative problem of synthesizing too few globin molecules, while the latter is a qualitative problem of synthesizing an incorrectly functioning globin. Alright. तो बाद में सिंपल सेल में क्या हो रहा है, क्वालिटेटिवली हम कुछ और ही बना रहे हैं ऑल्दो, राइट, और यहाँ पे क्या हो रहा है, there's a few globin molecules produced in case of thalassemia. It's a quantitative problem. Sickle cell anemia is a qualitative problem in case, although both are just anemia related. Chromosomal disorders में आते हैं, on the other hand they cause absence or excess of abnormal arrangement of one or more chromosomes, ठीक है. अब arrangement ही chromosome का गलत है. Failure of segregation of chromatin during cell division cycle results in the gain or loss of a chromosome. ठीक है, completely segregate नहीं हो रहा, it's called aneuploidy. हाँ, या तो क्रोमोसोम बढ़ रहे हैं या तो कम हो रहे. फॉर एग्जाम्पल डाउन सिंड्रोम, दैट इज अगेन एक्स्ट्रा कॉपी ऑफ क्रोमोसोम 21, पूरा का पूरा क्रोमोसोम ही ट्राइसोमी ऑफ 21. ये आप याद रखते हैं. तो क्या हो रहा है डाउन सिंड्रोम में, बेसिकली extra chromosome are. Similarly, Turner syndrome is due to a loss of an X chromosome in human females. Failure of cytokinesis after telophase stage of cell division results in an increase in a whole set of chromosomes in an organism, and this phenomenon is known as polyploidy. Polyploidy और एनीप्लॉइडी में यही फर्क है कि एनीप्लॉइडी में एक आधा क्रोमोसोम गेन होगा या लूज होगा, लेकिन अगर साइटोकाइनेसिस ही नहीं हुआ टेलोफेज के बाद, डिवाइड ही नहीं हुई सेट, तो पूरा का पूरा सेट जो है वो इंक्रीज हो जाता है, तो उसको पॉलीप्लॉइडी बोलते हैं. Helpful, but I think polyploidy, you see it in past, but in animals it's lethal. Total number of chromosomes in a normal human cell is 40 to 23 pairs. Out of these, 22 are autosomes, one pair are sex chromosomes. Sometimes, though rarely, either an additional copy of a chromosome may be included in an individual, or an individual may lack one of any one pair of chromosomes. These situations are known as trisomy or monosomy of a chromosome. Respectively. Such a situation leads to very serious consequences in the individual. Down syndrome, Turner syndrome, Klinefelter syndrome are common examples of chromosomal disorders. Down syndrome में देखेंगे, because of this genetic disorder is the presence of an additional copy of the chromosome number 21, trisomy of 21. This order was first described by Langdon Down in 1866. The affected individual is short statured with small round head, furrowed tongue, and partially open mouth. Palm is broad with characteristic palm trees. Physical, psychomotor, and mental development is retarded. Klinefelter's में क्या होता है? Genetic disorders also cause the presence of an additional copy of X. तो Klinefelter's में X का एक extra copy है, resulting into a karyotype of 47XXY in males. Such an individual has overall masculine development, however, the feminine development of breast, that is gynecomastia, is also expressed. Such individuals are sterile. तो Klinefelter's is for males, एक extra X copy होगा, तो 46 की जगह 47 होंगे again. This is aneuploidy. और ऐसे इंडिविजुअल में ओवरऑल तो मैस्कुलिन होते हैं, but they're sterile and they also have gynecomastia. Fine. Alright, it's again फीमेल्स में होता है दिस फीमेल्स में यूजुअली दो होने चाहिए, एक्स एक्स होना चाहिए, एक ही एक्स क्रोमोसोम है, दैट्स 45 with X zero. Such females are sterile as ovaries are rudimentary besides other features including lack of other secondary sexual characters. Fine. The Turner syndrome for your females. Is it clear to everybody? I just hope is able to do a little bit of justice with this lesson, though it's something that's really mathematical, and you've got to have your pen and a copy in your hand and go deep into it to understand. I think for the ones jinke basics clear the, for a quick revision session, I can't even take it all the way to for like hours altogether. It'll be a problem for you only because this is for the ones who want to do one shot it really at the last moment. So I actually want it to be a last moment revision that you're just done with all the lines of ncrt for your exam. So I just wish you all the best for all the exams that are coming, and just stay motivated and stay with your books. I don't know at this point of time, and I know it might get a little harder. Obviously, when you have your date sheet out, or obviously when you know that your exams are actually nearing and with every passing day, but you just have to keep calm, and I think studies would be the better solution. Um, and I just hope you all good luck, and I'll see you in my next one. Thank you for watching.